We will start in about 5 minutes. Right, so I'll start by sharing my slides. Welcome to the 9th live session of structural biology. And today we'll be talking about different ways of visualizing biological macromolecules. So why do we need molecular visualization? So science learning is abstract, especially when dealing with what we cannot see. Computer modeling has provided a window to the unseen and transformed science research. So we had talked about the different levels of organization, from atoms to molecules to cells to organs and organisms. But molecules and atoms are not visible to the naked eye. However, they can be observed by computer modeling, and this enables us to picture what previously could only be imagined. Before these visualization softwares were developed, molecular structures were presented by physical models of metal wires, rods, and spheres. For example, this is the first X-ray crystallographic solution study of a macromolecule myoglobin that Kendrew and co-workers had built. They had used brass models at a scale of 5 centimeter per angstrom. So per angstrom in the protein was translated to 5 centimeter in the model. The models were built and supported by vertical rods. But such a large physical model, carrying it around and transporting it becomes very cumbersome. So computer hardware and software technology were developed, as well as computer graphics program, which made visualization and manipulation of three-dimensional structures possible. Using these computer graphics, protein structure can be compared to understand the functions of proteins. And such visualization helps scientists and biomedical experts to study, understand, and engineer biological macromolecules. So, different protein structures can be compared using these software, and we can find out the active sites of proteins and even predict the mechanism by which they work. First, we will talk about the earlier methods of molecular visualization before all this software existed. So one of the first methods was called Richards box or Fred's Folly. This was a visualization device built by the biochemist Fred Richards in 1968, and it was known as the optical comparator. Electron densities resulting from crystallographic solution of the compound was calculated by computers. Then half-silvered mirrors were arranged in a diagonal way as shown here to superimpose these electron density maps upon a brass protein model. So the electron density maps that were calculated by the computer were held like this. The physical electron density maps were held in vertical rows according to like this. The surface level electrons were represented by the first map. And as we go deeper into the protein or macromolecule, the other level electrons were denoted by the other vertically placed maps. These maps were superimposed by this half-silvered mirror onto a brass model of the compound, and so the electron density could be denoted on the compound itself. As the larger molecules were solved, the scale was reduced to 2.5 centimeter per angstrom and then 1 centimeter per angstrom. So basically, as larger and larger molecules were solved, the number of centimeter that each angstrom translated to was reduced so that the model can be of a reasonable size. After this, there were physical ball and spoke models where each atom was represented by a ball and the bonds were represented by spokes. So after Kendrew et al. Had built a model like this using brass rods, they had then built a ball and spoke model of myoglobin, where each atom and bond could be observed. Another method was called Bidon's bender. So Bidon Rubin invented a machine for bending wire to follow the backbone trace of a protein. So what this machine does is that it allows the wire to be bent in whatever direction is needed, and the net orientation of the wire denotes the backbone of the protein. These small backbone wire models from Bidon's bender were the most manipulable and portable models available at the time. So compared to the ball and spoke model or the model derived from the Richards box, the Bidon's bender model, since it only consisted of a thin wire, was much more portable. Then came molecular sculptures. These sculptures were done in various media. For example, Bathsheba Grossman rendered these proteins in glass blocks. He would make laser crystals in which the macromolecule is represented by tiny laser-induced fractures within this glass block. Other people have also made sculptures in metal or wood. For example, a metal structure of an alpha helical protein is shown here. In the late 1990s, efforts began independently by Michael Bailey and Tim Herman to use new engineering technologies of rapid prototyping for building physical models of proteins. So basically what they did, they first created the mold for the protein using wax, and then using this mold, they built the protein structure and colored it according to, say, different secondary structures, or whether the residue was in the external or internal of the protein. So this was rapid prototyping, in which using the same mold, several proteins of similar structure could be created. Next came the generation of computer representations. So the earliest computer representation was in 1964, when Cyrus Levinthal and his colleagues at MIT developed a system that displayed on an oscilloscope rotating wire frame representations of macromolecular structures. So this was the initial computer representation that they had found, where the entire macromolecule was represented by a thin wire diagram, and it could be rotated in different directions. After that, the ORTEP representation was found, where in 1965, Carol Johnson released ORTEP, which was a program to produce stereoscopic drawings of molecular and crystal structures. So ORTEP stands for Oak Ridge Thermal Ellipsoid Plot Program. And it created structures like the one shown here, where each atom can be was shown clearly, as well as the bonds. And the stereochemistry was also clearly observed. ORTEP rapidly became a favorite of small molecule and protein crystallographers to produce illustrations of structures for conference presentations and publications. The most well-known use of ORTEP was by C.K. Johnson, who produced stereoscopic drawings of myoglobin, vitamin B12, and polyalanine using ORTEP. So these were the alpha helical structures of myoglobin that had been generated using the ORTEP program. One of the large advantages was that the computer kept track of the atomic coordinates, whereas atomic coordinates had to be measured manually from a Kendrew model, atom by atom. Just excuse me, I think someone wants to join. I think there is some network problem with on his side, but anyway, this lecture will be recorded, so you can always look it up on YouTube. The link will be provided. So right. Okay. So I'll continue with the lecture. So as I was saying, the ORTEP representation was uh, found in 1965, and one of the best advantages of it was that the computer kept track of the atomic coordinates, whereas the atomic coordinates had to be measured manually from a Kendrew model. So we had looked at the models that Kendrew had made, like this rod model or the physical ball and spoke models. So the problem with these models was that the XYZ coordinates of all the atoms had to be manually calculated and recorded. But when the computer representations such as ORTEP came into use, all these atomic coordinates were maintained by the computer itself. The next generation of computer representations consisted of MolScript, which was a program for creating schematic or detailed molecular graphics images from molecular 3D coordinates. Like you can see on the right side here, these are the MolScript representations of different secondary structures of a protein. So basically, the users had to supply an input file which specifies the XYZ coordinates, and the MolScript program would render images like this, which clearly showed the folds and secondary structures. For example, in this protein, you can clearly see this is an alpha beta barrel where there are beta sheets in the middle and alpha helices on the periphery. This is a bundle of alpha helices. And so like this, the MolScript program could represent the different protein structures. Next was called the persistence of vision or POV ray visualization. This is a cross-platform ray tracing program that generates images from a text-based scene description. And this ray tracing occurs from the camera to the scene. So basically, if you imagine a stack of atoms like this, what this POV ray visualization does is that it imagines a ray of light from the camera onto this stack. So you can look at the stack from different angles and you will get different pictures like this shown on the right. So like if you compare all the different pictures that you get from all the different ways of visualizing this three-dimensional object, you can get an idea of what the actual 3D object looks like. So the POV ray visualization is based on a single direction of visualization. So if you look at from the top, you will get a different image. If you look from the side, you will get a different image. It is only when you compare and correlate all the images that you can get an idea about the net 3D structure. So to help with understanding and learning the computer representation, something called TAMS, which stood for Teaching Aids for Macromolecular Structure, was brought out. So what people were seeing that it was becoming very expensive to publish an atlas of computer generated space field images in color. So what was found that there is a cardboard viewer could be used to look at stereo slides. So basically, these two are called stereo view images of the protein. So if you look at it, it looks like a two-dimensional representation. But if you look at this picture through this stereo viewer, then these two will superimpose and show you a three-dimensional structure of the protein. So obviously I can't demonstrate that through the slide, but this stereo viewer is available, and it is used mainly to convert two-dimensional images on paper into a 3D image, which helps understand the orientation and stereochemistry of the proteins. So like for example, suppose there are two trees, one behind the other. If you look at it from only one hole of the stereo viewer, you will not understand the relative orientation properly. But if you look at it from both the holes of the stereo viewer at the same time, then the images created in each eye will superimpose and give you a proper idea of the three-dimensional orientation of the biological macromolecule. Then different computer systems came into use in order to help with the computer visualization of the different biological macromolecules. The earliest was the Evans and Sutherland computers from 1980 to 1990, and these cost around $250,000 at that time. What these computers could do is that they could display the electron density map, and from the electron density map, you can fit an amino acid sequence manually, which matches that of the map. For example, suppose here you have a large electron density, and that can correspond to a pi electron ring like that present in the tyrosine side chain. So from the electron density map, manually the amino acid sequence can be fitted. Next came David and Jane Richardson's KindMages in 1992. So David and Jane Richardson, they pioneered this computer graphic representation of molecular structure with a series of programs. KindMage or kinetic image are set up to illustrate a particular idea about a 3D object. So suppose we want to find out what is the possibility of this 3D structure being the correct one. So using kinetic image, you can find out that how many Ramachandran outliers there are. That means how many residues in this protein are not falling in the allowed region of Ramachandran map. So that would mean that those residues in the protein, probably the prediction for those residues is wrong, because as you know that generally at least 98% of the residues have to fall within the allowed region of the Ramachandran map. Similarly, this kinetic image or KindMage can give you an idea about whether the structure you have predicted is good or bad. So it can predict the amount of steric clashes which would not favor the structure, or the rotamers which make the structure unfavorable. So kinetic image basically gives you an idea about the kinetics of the structure that you have predicted, and whether the kinetics are allowed or whether they are not favorable due to steric clashes or Ramachandran outliers. Next came RASMOL, which many of you probably use in your lab. So there was a need for a program to allow self-directed, neutral, interactive exploration of any available structure. So the problem with KindMage was that it is not an unbiased opinion. For the KindMage program to run, the user has to import certain criteria that biases the program to show what the user wants to see. So there was a need for a neutral, independent program, and that is what RASMOL provides. So RASMOL also provides a much richer sense of rendering, including space filling models. So you can see this is a space filling model of a protein interacting with DNA. And also the options for shadowing and reflective surface was available. So you can, in this protein, you can understand that this residue that suppose this lysine 7 residue is at the top part of the protein. But say this lysine 38 is present behind the lysine 7. So basically, RASMOL is giving an idea about the depth perception of the protein, which residues are on the surface, which are in the interior, can all be understood by RASMOL. Further improvement in the speed of the shadowing algorithm made the interprocessor communicator rate limiting on parallel processor computer. So what this means is that initially the computer processor that was needed for RASMOL used to be huge. So there a lot of parallel processors were needed, and to fit all that, it would require a large area. But with improvements, the RASMOL can now be implemented in personal computers or laptops on Windows and Unix. So there has been a large improvement in the processing power of RASMOL. And this RASMOL is widely used throughout the world by students as well as researchers, because as you can see, it among the different computer systems we have seen so far, this gives the best idea about what is happening in reality, because it is the three-dimensional image generated by RASMOL is very close to that what exists in reality. So we have studied about the protein data bank. So for the protein data bank, if you want to find a particular PDB file, you have to type the 4-letter PDB code into the search box. So I'll demonstrate that now. So suppose this PDB code 3HP1. I want the PDB file corresponding to that. So I'll just, in the Google search bar, you just type protein data bank. And then you will get this homepage of the protein data bank. In here, there will be a search bar, as you can see. Here you have to type the PDB ID. So we are searching for, one second, 3HP1. So you have to type 3HP1. And you will get the protein structure, as you can see, for the 3HP1 PDB. So all the details will be mentioned here, that what is the organism, Homo sapiens. So it is from human, and the expression system is E. coli. The date where it was deposited, please allow whoever wants to enter right now. So the date it was deposited and released, everything is mentioned in the PDB. So there is detailed information about the structure with that PDB. To display the different features related to the PDB file, we have to click. So we have to first download the PDB file in order to study it. So I'll once again open it. There is this option, download files. You have to, display files, and you can see there are different formats in which you can display. So suppose we want the PDB format. So the PDB format, you cannot, I'll go to the different formats in the next slide. So there are different formats in which you can see the files. The first is called the FASTA format. It is basically a text-based format for representing either nucleotide sequence or amino acid sequence. So in the protein, whatever is the amino acid sequence will be shown with the single letter codes of the amino acids. And if it is a DNA, then the nucleotide will be shown with the single letter nucleotide representation. So that forms the FASTA file. And in the original format, the sequence is represented as a series of lines. As you can see, there is many lines, and each line has a maximum of 80 characters. The format also allows for sequence names and comments to precede the sequences. So here you can see the sequence name is given, and any comment about, say, which organism it is taken from can be written before the sequence. And for writing this name and comment, a greater than sign is given. And also at the end of the sequence, there is an asterisk or star. The simplicity of the FASTA format makes it easy to manipulate and parse sequence using text processing tools and scripting languages like Python, Ruby, and Perl. What this basically means is that since the FASTA format is very simple, it just shows the sequence of amino acids. Then suppose you want to find a protein which has the amino acid sequence leucine, ala, valine. Then using these programs like Perl programming, you can search that in the protein data bank, that which proteins contain that particular amino acid sequence, and the Perl program will give you the output of the PDBs that contain that sequence. So if we go back to the PDB file that we were seeing, which is 3HP1, we can go to display files here and then FASTA sequence. So you can see the amino acid single letter codes are given. And in the first line, the name of the PDB, that is 3HP1, and the name of the protein, deoxycytidine kinase, and the organism it is taken from, Homo sapiens, is given. So for writing this comment line, a greater than sign is given. The next file format is called a CIF, or crystallographic information file. So in the CIF file, this is a standard format proposed by the International Union of Crystallography, or IUCN. So in this format, basically, different characteristics of the crystal is given. Like the symmetry cell is said to be triclinic, or the space group that is shown here as P1. And also the different cell parameters, like the side length A, B, C, or the angles alpha, beta, gamma of the single unit cell. So all these details about the crystal is given in the crystallographic information file, or CIF file. Then the next information is that for each atom, the XYZ coordinates are given. So we'll just open the CIF file. This is our crystal structure. If we go to display files and see this CIF format, you can see that the initial is all the information about the journal that this crystal structure was reported in, the pages of that journal, the author, all that is given. So these initial information is not that important. What is important is what I'm highlighting here. So this is the unit cell parameters. So the length of the sides of the unit cell and the angles. Then you can see the space group is given as P4321-2. So the details of the crystal is given, and that is why it is called a crystallographic information file. A further improvement of the CIF file is called a macromolecular crystallographic information file, or MMCIF. So in this, in the normal CIF file, all this information about the journal author is not given. But in the MMCIF file, all this additional information is included. Like I showed the journal name and the journal page number, and all that is present in the MMCIF file. So if we look back, this is actually the more advanced form of the CIF file, or the MMCIF file, where you can see the journal is biochemistry, the volume 48. So all that information is given. The next file format is called an MTZ file. So in an MTZ file, what is its significance is that an electron density map can be read from an MTZ experimental data file. Now loading this MTZ file is slow because the electron density map needs to be calculated. So in the other file formats that we saw, like the FASTA or the CIF, all the information is already there. So this file, those files can be generated very rapidly. But for the MTZ file, this electron density has to be calculated, and so generating it is pretty slow. But what can be done to speed it up is that you can get a rough electron density idea and later refine the electron density as you get more data. So when reading an MTZ file, you will get an option like this as shown here, and you have to mention the structure factor and phase. Because if you remember, we had studied in X-ray crystallography that only when you know the structure factor and phase can the electron density be calculated. So once you give that data and you mention the grid size. So grid size means you can see these blue lines forming a grid. So the grid size is the distance between these blue lines, and it is generally set at 0.75 angstrom. So you can definitely set it at higher resolution, but then the generation of the electron density map will become even more slow. So once you give the structure factor and the phase information and you mention the grid size, then this electron density map can be generated. And this can be done for any PDB structure reported in the protein data bank. The next part that is important is the validation report. So validation report, if I'll show, so we had seen the 3HP1 protein. So this is called the validation report, where it is mentioned that Ramachandran outliers means how many residues are not falling in the Ramachandran allowed regions. So, and then side chain outliers are, suppose the main chain of the residue is falling in the Ramachandran allowed region, but the side chain of the residue is in the Ramachandran not allowed region. So that is the side chain outliers. So there are different scores like this, which measures whether the crystal structure is good or bad. So if this and this bar that you see, the solid colored bar is comparing is how good this crystal structure is with relative to all X-ray structures. And this white bar is how good this crystal structure is with relative to X-ray crystal structures of similar resolution. So as you are more towards the red, this is not a good crystal structure. And if you are more towards the blue, this is a good crystal structure. So this is called a validation report. So if you see this slide, this is comparing between a good and a bad structure. So in the good structure, most of these points are towards the blue end of the columns, which means it is a good structure. Whereas in the bad structure, most of these points are towards the red end of the spectrum, and so it is a bad structure. These sliders report on how good the structure is relative to all the structures present in the PDB. So, and here you have another, so this is comparing the whole structure with other structures. But if you want to see a residue-wise validation, then a residue sequence plot is also available, where any residue which has unusual geometry are color-coded. So the residues which are green over here means that they are perfectly fine. They are not outliers. If as the color goes from yellow to orange to red, that means those residues become more and more disallowed. So in this good structure, you can see most of, there are only one orange and no red residues. So the residues are mostly allowed. But in this bad structure, you can see there are multiple orange and even one red. So many of the residues are not allowed. For X-ray crystal structures, a red dot indicates a poor fit to electron density. So if there is a red dot above the structure, that means the structure that you have predicted is not fitting to the experimental electron density. So in this good structure, there are very few red dots. That means the predicted structure is good. But in this bad structure, there are so many red dots. That means the predicted structure is not very good and it can be improved much more. So this validation report is very important to check the validity of a structure deposited in the protein data bank. So how do you read a PDB file? Each column, each line is 80 columns wide and is terminated by an end of line indicator semicolon. So you can see for each line, there is a semicolon at the end. The first 6 columns of every line contain a record name. So header, you can see this word header, that is called a record name, or title, compound. So whatever is present in the first column, that is called a record name. And these have to be exactly the same. This first column has to be exactly the same in all PDB files. So in the PDB file, you can see in the first line, the type of molecule will be mentioned, which in this case is a transferase. The date at which this data was released will be mentioned, and the PDB ID will also be mentioned. Then if you go down, you can see it's written molecule. So this means this is the name of the molecule, which over here is deoxycytidine kinase. The chain number and name is also given. The scientific name of the organism is given, and the common name is given. So the scientific name Homo sapiens is mentioned, and below that, the organism common name human is mentioned. Then the author details will be given, that who are the authors who have deposited this structure, and the resolution of the structure will be mentioned, which in this case is 2.31 angstrom. The next important thing is this record, which is called CRIST1. So you will see that in one of these lines, as I'll just show you. So if you go to display files and you take PDB format. So this is the PDB format. So as I was talking in the beginning, the name of the molecule, the name of the authors, the journal, everything is mentioned. And if you eventually go down, suppose I'll search so wherever this is written, CRIST1, that is the details of the crystal structure. So if you look back, this is the unit cell of the crystal structure. So the crystal structure has 6 unit cell parameters: A, B, and C, which is the length of the unit cell in the X, Y, and Z directions, and alpha, beta, and gamma, which are the angles. So these 6 parameters will be mentioned, and the space group will also be mentioned. If by chance, say the structure in the protein data bank was not determined by crystallographic means, then for this record, it will just be a unit cube. So the default is a unit cube. So suppose someone has deposited a structure which they found by NMR. So then all these values will correspond to that of a unit cube, since that is the simplest crystal structure. Next, if you see wherever atom is written, that will mention the atom number and the atom symbol, which is nitrogen. Ca stands for C alpha, Cb for C beta, the residue to which the atom is belonging, and the chain number is also mentioned, and this is the residue number. Then the X, Y, Z coordinates are given, and the occupancy is given. So suppose occupancy of 1 means the position of that atom is fixed at that position. If the occupancy was 0.5, that means the atom is existing in that position for only 50% of the time. Then the thermal factor is given, and finally the element symbol. So all this data you can find from the PDB file. finally, at the end of the PDB file, if you look back, so if you search actually for, you can see this TER. There is a word TER that is written. And TER shows the termination of that particular amino acid chain. It represents the carboxy terminal residue, meaning the C-terminal residue. Then in many cases, instead of atom, you saw atom was written here. So in many places, het atom will be written. So het atom generally stands for atoms which are outside of the amino acid chain. For example, if there are water molecules, that will be written, het atom will be written for the atoms of the water molecule. And at the very end of the file, I'll once again show the PDB. So at the very end of the file, the end word has to be written, END. If you look at, suppose there are water molecules surrounding the structure, then they will be represented by het atom. And many times there is this record called connect, CONECT. So what this does, it defines the different interactions. So if it is connect and then two values, that means there is a bond between these two atoms. So these are the atom number, and it means there is a bond between these atoms. If it is connect and then four values, that means there is a dihedral angle between these four atoms. Then if it is connect and three values, that means there is a normal angle between these three atoms. So this word connect, it basically indicates which atoms are connected either through bonds or through dihedral angles or through normal angles. So from all the information that you are getting from a PDB file, different parameters can be calculated. So one of these is the distance between two atoms. So as you saw, for each atom, you can find out the X, Y, Z coordinates from the PDB file. Once you know the X, Y, Z coordinates of two atoms, 1 and 2, if you put it in this formula, you can find the distance between these two atoms. So from the information in the PDB file, you can find distance between any two atoms. Similarly, between three atoms, you can calculate the angle theta. Again, all this requires is the XYZ coordinates of all the three atoms. And that is very easily available in the PDB file. So once you know the XYZ coordinates of all the atoms, you can just put it in the formula and find the angle between the three atoms. The same concept is used for dihedral angle. So in dihedral angle, the calculation is a bit more complex because it involves cross product and dot product of vectors. So, but all the data that you need is the coordinates of these four atoms that are connected in the dihedral angle. So once you have the coordinates of P1, P2, P3, P4, you can just put it in the formula and then get the cross product and dot product respectively and find this dihedral angle between these four atoms. Similarly, the data from a PDB file can be used to predict that what is the secondary structure in the molecule. So we had learned about alpha helices, and we know that in alpha helices, there is a hydrogen bond between the ith and I plus 4th residues. So from the PDB file, you can find out the distance between the carbonyl and the NH of the ith and I plus 4th residues. If this distance is less than 3.5 angstrom, then a hydrogen bond exists. And if you find multiple such consecutive I plus 4 to I hydrogen bonds, that means it is an alpha helix. So once more, what you need to do is that you need to find out the atom numbers of the CO and NH. Then the PDB will give you the XYZ coordinates. Using this formula, you can find the distance between CO and NH. And if this distance is less than 3.5 angstrom, that means there is a hydrogen bond between those atoms. So if there are multiple such consecutive hydrogen bonds between I+4 to I atom residues, that means it is an alpha helix. Similarly, you can find out whether the secondary structure is a beta sheet. So in the beta sheet, residue I in strand 1 forms a hydrogen bond with residue J in strand 2. The next bond can be formed in two ways. The residue I+2, the residue I+2 can bind to J+2, which will form a parallel beta sheet. Or the residue I+2 will bind to J-2, which will form an anti-parallel beta sheet. So once more, how you can find this is that you first identify the CO and NH atom numbers, and the PDB file will give you the coordinates of these atoms. So using the XYZ coordinates, you can find out the distance, and if the distance is less than 3.5 angstrom, there is a hydrogen bond. Now, if the hydrogen bond is like this, the case that is shown in this first example, then it is an anti-parallel beta sheet. And if it is like this, as shown in the second example, it is a parallel beta sheet. So the secondary structure can be predicted from a PDB file. So just give me 2 minutes. So now we will learn about one of the molecular visualization softwares that is called COOT, or Crystallographic Object-Oriented Toolkit. COOT is for macromolecular model building, model completion, and validation. It is particularly suitable for protein modeling using experimental structure determination data. So COOT displays electron density maps and atomic models and allows model manipulations. Suppose this is showing an output from COOT where the electron density map is shown by this blue mesh, and the possible orientations of the atom are shown with this yellow and this gray line. So as you can see, this gray orientation is matching with the electron density map. So that must be the correct orientation. So COOT displays these electron density map and atomic models and helps to get a more ideal model of the structure. So suppose in the less ideal model, we had initially predicted that this side chain would be oriented in this direction. But from the electron density map of COOT, you can find out that it is actually the other orientation in which the structure and the electron density map are matching. Other than that, rotation, translation, rigid body fitting, Ramachandran plots, and all can be done using COOT. So if you have a protein and you want to find out if it is Ramachandran allowed, then COOT helps you plot the phi values of the protein residues on the Ramachandran map. As you can see in this particular example, most of the residues fall within the allowed regions of the Ramachandran map. And so clearly, this is a valid protein structure. There are very few outliers. So you can see that in the preferred regions of the Ramachandran map, almost 94% of the residues are falling. Only around 5% of the residues are in the non-preferred but still allowed regions. And only 2 outliers are present, shown by the red dots. So clearly, this is a very good protein structure. This software is very easy to, so you can download it for free. So you can just type type install crystallographic object oriented toolkit and you can download it for free. And it is very easy to learn for even new users because all the tools are very easily discoverable. That is, the user interface is very friendly. So as I said, Coot is free software and it is available from the Coot website, originally at University of York, and now it is at the MRC Laboratory of Molecular Biology. So these are some of the uses of Coot. So basically, Coot can be used to read files containing 3D atomic coordinate models of macromolecular structures in a number of formats, including PDB and MMSIF that we just studied. The model can be rotated in different in 3D and viewed from any viewpoint. So I'll just show you. So I had installed Coot. So I'll just open it and show you. Okay, so you need to go to File, Open Coordinates, and then open a PDB file. So I'll open one PDB file and show you. Actually, I'll start from downloading the PDB file itself. So, so you can, I had shown, this is the PDB file, and you have to go to Download Files and then PDB format. So now the PDB file is downloaded. I'll just copy paste it onto the desktop so I can easily show you how Coot works. Yes, so this is the window of Coot. You have to go to File, Open Coordinates, and choose the PDB file. So you can see this is our PDB file, 3hp1.pdb, and open. So this option will come that there are some nomenclature errors. You type yes to correct them. So this is showing the 3D view of the molecule. You can rotate it in any direction that you want, and you can see the protein. So, and you can enlarge and reduce the size also. So if you want to see a particular area, you can enlarge it and move the protein as you want to observe it properly. So coming back to the presentation. So you can rotate the model in any direction, and it is generally represented by a stick model. So the atoms are represented by sticks, and wherever there's an oxygen atom, it is shown by red, and the nitrogen atom is shown by blue, and the carbons are this yellow. So that is the default coloring that is used in Coot. Coot can also display electron density, which is the result of structure determination experiments, such as X-ray crystallography and EM reconstruction. The density is contoured using a 3D mesh. So if you go to, actually this calculation will take some time. So I had done it previously, so I'll just show it here. So this blue mesh that you can see is showing the electron density, and this can also be done in Coot. So there are many features that it provides for model building and refinement, meaning if you want to fit the model to the electron density better, Coot can provide options for that, and also validation of the model, that whether the predicted model is fitting with the experimentally observed model. So you can download this software for free, and there are many tutorials available. I don't have the time to go into all the features, but you can easily look into the different tutorials of this software. So I'll just continue. So one of the applications of COOT that has been used recently is that for looking at the mechanism of beta lactams as antibiotics. So we had learned in one of the previous classes that beta lactams, so beta lactam is basically a four-membered ring which has a NCO bond. So this bond, it can be broken by a beta lactamase enzyme, which will break the amide bond, forming a NH and COOH. So when the beta lactam ring is present, this can act as an antibiotic. But when this ring is broken, it becomes inactive. So what was done is that the PDB and MTZ files of the beta lactamase complexed with the beta lactam before this reaction and after this reaction was taken. And the change of conformation was identified using COOT. So this change, so if you study the change of conformation, then this mechanism was found where initially this serine OH is attacking the amide CO, and this amide bond is breaking open. So this serine O is attacking this double, and this NCO bond is breaking, becoming NH, and serine is attached to this side. Then once again, this nitrogen, basically it is rearranging. The double bond is rearranging, and you can see in the final product, there is a deacylation. So this mechanism was predicted using COOT, and it was understood that how the activity of antibiotics can be deactivated using beta lactamase. So as we discussed, COOT is very easy to use and it is a powerful interface, and it provides opportunity to do functional analysis. So building a model, refining the model so that it fits better with the electron density map, and analyzing the electron density map is making COOT very popular to the structural biology as well as computational biology community. So just here, if you see that what is the effect of the beta lactam. So bacterial cells, cell walls have a protein called peptidoglycan, which is basically a peptide complex with a sugar. And if it is exposed to penicillin, which is a beta lactam, then it breaks the peptidoglycan in various points. And then the cell wall, which was initially very strong, it becomes weak wherever the peptidoglycan is missing. Then if you expose this to a hypotonic environment, suppose dilute, meaning environment that is more dilute compared to the environment inside the cell, then the water will diffuse into the cell and the membrane will bulge. And eventually it will reach a breaking point when the bacterial cell wall breaks completely and the bacterial cell is destroyed. So this is how the penicillin works. However, many bacteria have evolved now to have beta lactamases, which make the penicillin inactive by breaking the beta lactam ring. And that is why many bacteria are now developing antibiotic resistance. A more popular molecular visualization platform is PyMol, which probably many of you use. So PyMol is also free to download. So I'll show you the working of PyMol as well. So what you can do, so there are two ways to open a PDB file in PyMol. Firstly, one second. So PyMol is here. Sorry, not this. PyMol it's here. I think I'll close COOT for now. Yes, so this is the PyMol terminal. So you can go to File, Open, and choose the PDB. So we had 3HP1. Yeah. Then open. So this is the PyMol structure that you can see. And another way to do it is, I'll just close it again. Another way to do it is to type a command. So the command is called fetch, 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 and then the PDB ID. So this is, there is a bar here where you can type the command, and you have to write fetch 3HP1. And you can also get the PDB like that. So suppose you have the PDB file downloaded, you can just open it. Or if you know the PDB ID and you have not downloaded the file, then also you can open it by typing the command fetch and the PDB ID. So next, suppose I want to change the color of the protein to this green color that is shown. So for each color, there is an RGB value, meaning red, green, blue value that you can find out. And there are certain commands that you can use to change the protein color. So the first command, I'm just copy pasting from this PPT. You again have to type it in this command bar that is there. So the first command is set underscore color space. You have given the name of the color, comma, and in third brackets, the RGB values. So and then enter. And then you have to give the second command. So that particular RGB value is for that dark green color. So this second command, if you now give, see the entire protein becomes dark green in color. You can easily rotate and see everything. So you can see the different ligands that are there in the protein and also the secondary structure. So here it is taking an alpha helical structure. And if you look at it here, it's a beta sheet. So you can see these arrows, parallel beta sheet it is representing. So now you have set the color of this protein to this dark green color. Next, if we want to see the electrostatics, we have to go to this wizard option, wizard, demo, and then electrostatics. So it is showing the so wherever red is there, that is negatively charged, and blue is positively charged. So suppose this protein is binding with a positively charged ligand, then then the ligand will bind to these red areas, which are negatively charged. Similarly, if it is binding with this protein does not have many strong positively charged areas. As you can see, the red is very intense, but the blue is quite light. So mostly, if we have to guess whether this protein will bind to a positive or negative ligand, most probably it will bind to a positively charged ligand because this red area is quite intense. So now we have seen how to open the PDB, how to change its color, and how to look at the electrostatics. If we go to the next slide. So now we will see how we can view the side chains. So I'll just close this for now. Yeah. I'll again open the PDB. Fetch 3HP1. You can see I've typed the command here, fetch 3HP1. And I've gotten the PDB. So now I want to look at the side chains of this PDB. So there is this command. Again, I'm copy pasting from this slide. So one thing you have to do is you have to One second. You have to first choose the residue. So if you can see at the right hand side of this slide, it is written selecting residues. That means it is now in selecting residue mode. So suppose I choose a random residue. I have chosen this residue, and then I will type this command. Once you have done that, you can view the residue by right click, show. So you click on the residue that you want to see and you right click and then show. And once you go to show, you select sticks. So you can see this is a tyrosine residue. Because you can see the side chain, you're seeing the side chain of the tyrosine residue. One second, yeah. So, you know, tyrosine is a phenyl ring with a OH at the para position. So here the side chain is clearly visible of the tyrosine residue. Then if you want to measure the distance between two residues, between two atoms, sorry. Suppose I will take another residue. I've selected the residue, then right click, show sticks. Okay, that is probably a glycine. So one second. Yeah, that is an alanine. So you can see only a CH3 is coming out. So that residue is an alanine. So if I want to find the distance between, say, this carbon of the tyrosine and this CH3 of the alanine, then what we have to do is we have to click on measure or wizard, then measurement. It's written, please click on the first atom. So I've clicked on this carbon. Please click on the second atom. I have clicked, say, on this carbon. So the distance is given in angstrom. If you can see, it's written 4.6. I think it's clear. So the distance between these is 4.6 angstrom, which is not close enough for an interaction. So there is no interaction between these two atoms. So like this, if you want to find out if there is a interaction, whether it is van der Waals or hydrogen bonding, you can find out the distance between two atoms like this. Now, suppose I want to save this image. If I want to save this image, I will now go to display, background, white. So it gives a white background. Generally, if you look at the images in different manuscripts or papers, it is always on a white background. Then you have to type the following command. What this will do is that it will give a high resolution image. So I've typed the command. So what this will do is, first it will remove the background. So you can easily paste it wherever you want. Then you can uncheck the transparent background by right click. So I can just right click here. And it is now high resolution. And then file, save image as PNG and save. So I'll go to file. One second. Yeah, you can export molecule or save, basically. Save molecule, say whatever, try. and you save it by whatever extension you want, whether you want to save it as a PDB file or whatever, that is up to you, and you can save it. Okay, there is some error right now. So oh, it is still in measurement mode. That's it. Yeah, anyway, so you have seen how to find the distances. So next we will see. So molecular visualization. If you want to view an oligomer, then okay, now I'll open an oligomer and show that how the oligomer can be viewed. Just 1 second. So I have a dimer here. I have opened that. So it is a dimer, but you can see that it is difficult to understand that this is a dimer because the two chains are of different colors. So if I want to make it sorry, two chains are of the same color. So I need to make it different colors. So for that, we have to choose the chain and color. So here you can see it is now at selecting residues. So if I click on residues, it will change to chains. Now it is selecting the chain. So now I'll select the chain, right click, color, by chain, and again by chain. One second. So you can remove one of the chains. So suppose you can select it and hide everything. So right now you can study it one dimer at a time. So instead of studying the whole dimer, you can study the monomer. You can see that it is made up of consecutive anti-parallel beta sheets. So these arrows represent beta sheets. So it is kind of forming a barrel-like structure, and the beta sheets are connected by turns. So you can, from the dimer, you have formed the monomer, and you can study it separately. So as you can see, the dimer was there. You can separately study the monomer. So before studying this alignment with other proteins, we'll just look into another thing. So we'll take this exercise. So firstly, this is mainly about mutating residues. So we will go to Coot and we'll open a PDB file. I'm opening the 3HP1 again. Then we'll follow these instructions on making mutants. Mutants basically means if I want to change only one amino acid, to some other amino acid, then that is called making a mutant of that protein. So we'll go to draw and then go to atom. So this draw option is there, and then go to atom option is there. We'll do that. Then what the instruction says is that choose the residue you want to mutate and click on apply. So suppose I want to mutate residue number 45. I'll click on apply. So it is now showing residue number 45 is a cysteine, because you can see it is written here CYS, cysteine. So on the right side of the window, there is something called simple mutate. So if you look at these different options, when you bring the mouse on the option, then it will show that what is that particular option. So this option, which I'm pointing at right now, is called simple mutate. You can click on that. So this plus sign appears. And if you click on the plus sign, 1 second, the plus sign will appear. You click on the residue and choose the amino acid you want to change it to. So suppose I click on this residue and I want to change it to, this is a aspartic acid. I want to change it to a basic residue, say lysine. It became a lysine. Did you see that? So again, if we want, we can just do simple mutate, choose the residue. This is a lysine that is there. I'll click on the lysine. I can mutate it to, say, a histidine. It now became a histidine. So like that, you can mutate individual residues and get a mutated protein. And after you do that, in PyMol, you can view the mutated files. So now that we have gotten this file, we can save it. This mutated protein that we have created, we can save it. File, save coordinates. Suppose I save it as trial 1. Save. So now I will open that mutated protein in PyMol. So this is the mutated protein that we have. And then I will get the original protein. So I'll write fetch 3HP1. Yes. So you have the original protein and you have the mutated protein and the original protein. So 1 second. You can easily compare their orientations. So yeah, this is so the original and the mutated proteins, and you can compare. Since we have mutated only one residue, this is the original, this is the mutated. So since we have mutated only one residue, you cannot see much conformational change, but you can see the different, suppose we had mutated many more residues in this alpha helix. Suppose we had mutated it to helix breaker residues like proline or glycine, then we would clearly see the change in conformation as we go from the mutated to the original residue. So PyMol can help you visualize that. There are many other functions of Qt and PyMol, but for now we'll restrict ourselves to this. Since these are all freely install, like you can download them for free, you can just look into some tutorials and go through different commands. So now that we have gone through the basics of this visualization of different biological macromolecules, I'll just go through some sample questions. So which file format is generally used for the amino acid sequence of proteins? I'll give you some time and then I'll discuss this. Right, so the correct answer is the FASTA file format. So if you remember, I'll go to the FASTA file format so you can see it directly. So in the PDB, if we go to display files and then FASTA sequence, you will see the amino acid sequence is given. So each amino acid is represented by the single letter code. So M for methionine, G for glycine, and so on. So since the question was that which file format is generally used for the amino acid sequence of protein, the correct answer is FASTA format or C. The next question is that the header line in a FASTA started with what? Meaning the first line where the organism name or comment is given, what does it start with? So the correct answer is C, which is a greater than sign. So if you remember, the first line before the single letter code is always a greater than sign, and then the name is given or the organism from where it comes, and then the sequence. So if we go to the actual FASTA file, you can see the first one starts with a greater than sign, and then there is the PDB code 3HP1, the chain number chain A, the name of the compound, and the species from which it comes. That is all in the first line, which begins with a greater than sign. The next question is that which of the following is not a protein structure visualization tool? Right. So the correct answer is VLC. So if we go back, we can see we had learned about PyMol, which is a molecular visualization software. We had also talked about COOT, which is a molecular visualization software mostly used for the electron density or the Ramachandran map to find out if the predicted structure matches that of the electron density that we get from experiments, or whether the protein falls in the Ramachandran allowed region, which means it is a correct structure, or if it falls in the Ramachandran disallowed region, which would make it an incorrect structure. And if we go back much further, we had also talked about RASMOL, where we had discussed that it was the first program where the 3D representation was shown with space filling models. So if there are different residues, like a residue on the surface like lysine 7, and a residue which is slightly behind like lysine 38, then that could be distinguished by RASMOL. So all three, RASMOL, PyMol, and COOT, are molecular visualization softwares. So clearly, since the question was that which of the following is not a protein structure visualization tool, the answer has to be VLC. The next question is that how many characters are present in the PDB ID? Right, so the correct answer is 4. So if we look at the PDB structure that we were studying, 3HP1 is the PDB ID. All PDB IDs like this have 4 characters. The first one is a number, and the next 3 can all be letters, or 2 letters, 1 number. But overall, it consists of 4 characters. So the PDB ID always consists of 4 characters. So the correct answer is B. Next, connect portion present in PDB files are used for what? which means that in PDB files, there is a word called CONECT or connect that is mentioned. So what information does that give? So the correct answer is D, or all of the above. So if you look at the PDB file that we have and go to display files and PDB format. So you can see there are so many this is called a reader. So this connect word is present in many lines. So what this is representing, when there are four numbers after the connect word, it represents a dihedral angle between these four. When there are yeah, when there are two atoms after this connect word, it means these two are connected by a single by bond. So there's a bond distance between these two. If there are four, that means there is a dihedral angle between these four. If there are three, that means there is a normal planar angle between these three. And if there are five, that also means they are connected kind of like a pentagon. So they are also connected in plane. So basically what this is showing, this CONECT, it is showing the connectivity between different atoms, whether there is a bond distance, bond angle, or a dihedral angle. So since the question was that what is this defining? It defines bonded interaction, angle, and also dihedral angle. So the correct answer is D, all of the above. The next question is, which command or tool is used for high definition images in PyMol? Yeah, so the correct answer is B, ray. So I'll demonstrate this right now. So I have this. I'll go to, first we have to make the background white. So display background white. Once you have done that, you can see this image is not very good, that it is blurry in some places. So you have to give a command that says which is this command, ray 1080, 720. So this ray command makes the image more high definition. As you can see, the image has become more high definition now. It's much more clearer. If you rotate it and see, it's much more clearer. So the command was here, it's ray or ray. So the correct answer that which command or tool is used for high definition images is B, ray. The next question is that which of the following is present in the model fit or refinement tool of code? Right. So the correct answer is simple mutate. Sorry. Uh, so if we go back, so simple mutate is used for refinement tool. How is that? So just wait a minute. We'll open quote. So suppose you have an electron density map and you want to find out what will fit better. So you have to mutate the residues to find out. So to do that, this is a phenylalanine residue. As you can clearly see, the phenyl ring. So I'll do simple, this is the simple mutate option. We'll click on the phenylalanine residue and I'll mutate it to say alanine. It became an alanine. So like this, you can change different residues and find out which best fits the electron density map. And that is why the simple mutate tool is used for fitting the model or refining the model. It is not Ramachandran map because Ramachandran map is to find out the validity of the model, whether the model is in Ramachandran allowed region or disallowed region. For fitting, the tool is simple mutate. The next question is that the measurement displayed between two atoms in pi mol has unit of what? Right. So the correct answer is angstrom. So if we open PyMol, sorry, it was already open. If we open PyMol and we go to wizard measurement and to find the distance between two atoms. So suppose I want to find the distance between this oxygen and this oxygen. Distance is showing 5.5. So the unit of this distance is in angstrom. So the correct answer is C, angstrom. The next question is that which of the following parameter one cannot get from the validation report from worldwide PDB? So the correct answer is D, macromolecule fold. So if we go to the validation report of the PDB, this is the validation report. You can see that entry composition. So what is, if you keep going down, scrolling down, you can see the composition of the protein, that where the molecule that it is, and the different amino acid sequences. So if you go to UniProt, you can find the amino acid sequence. It is showing the sequence matching with different other PDBs. You can see the different amino acids will be given. Then if you go back, so the composition is given, data and refined statistics. So this is the refined statistics where it is showing how refined the structure is compared to other X-ray structures in the protein data bank. Resolution and model quality. So resolution you can see is given here, and model is 2.31 angstrom, and model quality is described by the R values. So we have read about that. So model quality is also given. What is not given is the macromolecule fold. So if we, in none of the information here, can you find the macromolecule fold? So the correct answer is D, that you cannot get it from the validation report deposited in the protein data bank. The 10th question is that what is the format for the command of RGB color in PyMon? So the correct answer is C, which is third bracket, and the RGB values separated by commas. So if we again go back to where we had learned about this, there are two commands if you remember. So this is the first command that you have to give. You type the command in the command bar, and the RGB values are basically written in third brackets, and R, comma G, comma B, the integer values are given. And then you put enter. It is when you put the second command that the color will change. Now the color has changed, as you can see. So the RGB values are given in third brackets and they are separated by commas. So the question was that what is the format for giving the RGB values? It is C, third bracket, and A, comma B, comma C, where A, B, C are integers. The next question is that what is the important software to make diagrams? So the correct answer is all of the above. So you can use put to mutate different residues, and you can use PyMol to get high quality images. But why is PowerPoint needed? So basically, in papers, this is the format that is used. Suppose one residue has been mutated from glutamine to lysine. It is shown like this with a square, and then the square is enlarged to form a larger and expanded view of the region. So PowerPoint is needed to form these squares, these dotted lines, and to form the net image like this. So basically, you need all three softwares. So you need all three softwares: Qt to mutate the residue, PyMol to create a high definition image, and PowerPoint to assemble the image and create the dotted lines to show that it is an expanded image. So the answer is D, or all of the above. The 12th question is that what experimental structures cannot be deposited in PDB? So the correct answer is comparative modeling. So this we have learned about before, that structures deposited in PDB can come from X-ray crystallography, they can come from NMR, They can also come from mass spectrometry or cryo-electron microscopy, but they cannot come from comparative modeling, because comparative modeling is something that you do in a software. You take a protein that is already known and you take a protein which has similar structure to it, and so you can model the structure of the unknown protein based on the known protein. So that is a prediction that is done, and obviously that cannot be deposited in PDB. So the correct answer is C. The 13th question is that which of the following is incorrect about Swiss PDB viewer? So the correct answer is B. So it is a structured viewer for multiple platforms because it can view different formats like PDB zip format, and it is used for structural visualization, analysis, and homology modeling. So the incorrect option is B. The final question for today is, what is the general representation of a PDB ID? Where X is any integer and B and C, D can be alphabet or integer. This question is a bit incorrect. So B, C, D can be alphabet or integer. So the correct answer is A, which is X, B, C, D. So the first one is always an integer, but the next three can be alphabet or integer. So that is all the questions I have for today. And if you have any, you can check out the software Scoot and Pymol because they are free to download and there are many tutorials available. And if you have any doubts, you can email it to me, or you can ask them in the next class also. So that ends today's session, and I'll be back next week again on Monday.